Hi everyone. I apologize in advance because this project is going to be, or this talk is going to be a bit like an annoying abstract. I've left out a lot of the technical details because I recognize we have 3 hours to go through them in extreme detail tomorrow morning. So that's also a plug to attend the workshop if you're interested in learning more. With this talk, we really wanted to just give you an example of how eDNA metabarcoding is being used in the real world to help make decisions. And I think that'll offer a little bit of hope to the audience in terms of where this technology is going and can be used by people like myself who are predominantly ecologists who are doing impact assessments. So today I'm giving a talk called eDNA Metabarcoding as an emerging tool for informing biodiversity impact studies. And I'm going to give you an example of using this tool for informing a federally regulated impact assessment at a very early stage. So during this talk, I'll briefly go over the adaptive phase management project that eDNA metabarcoding is being applied to. I'll talk a little bit about the collaborative nature of a project of this breadth, depth, and purpose. And then I'll talk about the data collection and directions and where we're at right now. I'll also talk a little bit about how we hope that this project will eventually advance the use of this tool for impact assessments and for fundamental sciences in these areas. So the Nuclear Waste Management Organization was formed in 2002 as a requirement of the Nuclear Fuel Waste Act. The mandate of the NWMO is to achieve the safe and long-term management of Canada's used nuclear fuel in a manner that protects people and the environment for the long term. The NWMO was responsible for developing Canada's official plan, known as Adaptive Phase Management, for dealing with used nuclear fuel. The APM has been guided by an Indigenous Council of Elders and Youth, independent advisory councils, and participating First Nations communities and municipalities. Canada's APM emerged through a 3-year dialogue with Indigenous peoples and Canadians in general. During these conversations, some key priorities emerged. One, that safety of the project needed to be preeminent. Number 2 was that people generally agreed that we should be dealing with used nuclear waste now and not leaving it for future generations to deal with. Number 3 was that it should align with international best practices. And 4th, whatever approach is taken should be adaptable as new technology and learning happens. These priorities were encompassed by the APM, which includes both the technical methodology and a management plan. The APM calls for containment and isolation of Canada's used nuclear fuel in a deep geological repository located at a site with a suitable rock formation. The NWMO follows the principle that the project will only proceed in an area with informed and willing hosts, where the municipality, First Nation, and Métis communities, and others in the area will work together to implement it. The process for selecting a deep geological repository began with 22 communities that expressed interest in potentially hosting the project and learning more. The NWMO, through their work, has gradually narrowed their focus to 2 potential siting areas: the Wabigoon Lake Ojibwa Nation Ignace area, as well as the Saugeen Ojibwa Nation South Bruce area, both in Ontario. Currently, we are undertaking eDNA metabarcoding studies at both sites. But for this talk, we will be focusing only on the data collecting from the Wabigoon Ignace site in 2021 during the summer and fall seasons. The reason that we're focusing on these data only is we've spent the most time with these data and been able to work them out the most. Here I provide a bit of an IA briefer for those that are not operating within the impact assessment sphere. Once a site is selected, the APM will become a project of national and international significance. The project can only proceed following the federally regulated impact assessment and licensing processes that will be required. An impact assessment at the very highest level generally evaluates the overall costs and benefits of a project to society, which includes considerations of impacts to the social, economic, health, and environmental conditions of an area. Zoetica is responsible for the development and oversight of the biodiversity impact studies and assessment portion of the environmental pillar of this project. Canada's Federal Impact Assessment Act, which underlies the impact assessment process, is designed to contribute to sound and sustainable development decisions, to provide transparency and participation in decision-making. And now it requires that both Western scientific information and Indigenous knowledge is included in those decisions. So the general steps taken in an impact assessment, for those unfamiliar, is that first we define the environmental conditions or the baseline conditions of before the project is present. And this involves things like collation of existing information, collection of TK or traditional knowledge, and data collection from the area. We then use this baseline information along with project information to predict where impacts may happen. We then try to take steps to mitigate these negative impacts through the mitigation hierarchy, where first we try to avoid the effect. The simplest explanation of this is don't build there, don't build in this area. If that can't be done, we move through the mitigation hierarchy through steps of minimization, restoration, and eventually offsetting to try to reduce those negative effects. If those negative effects cannot be reduced through these steps, then it's our job to describe those effects and to assess them. Sometimes positive effects will also be residuals, and in those cases we try to enhance them. Where needed, we develop monitoring plans to follow up and to manage our uncertainty and to test the accuracy of our predictions. Finally, we apply adaptive management where it's required to keep impacts within the acceptable bounds. So during this talk, I am focusing only on the use of the very early adoption of eDNA metabarcoding at these sites as a tool for collecting baseline data as part of an impact assessment for the APM project. I'll be very clear that we do not advocate the use of this tool to replace traditional studies, but we hope to provide an example of where it's being used alongside traditional methods to help improve our knowledge about the biodiversity in an area. The eDNA metabarcoding studies that I will discuss required collaboration between many groups. This is a collaborative effort between Indigenous communities, the NWMO, conservation authorities, the University of Guelph, an independent peer review group, Zoetica, the company I am with, municipalities, and program technicians. This collaborative approach was deemed necessary early on as there is a huge breadth and depth of knowledge to make this program work for the desired purposes. We designed the field data collection protocols as ecologists to ensure that sites were sampled with little to no bias, such that data can be used to make generalizations about potential species found within various ecologically relevant study areas and habitat types. We elected to use a generalized random tessellation stratified sampling design, or a GRITS design, and we ensured that field and laboratory quality controls were included. We also planned for repeated sampling between seasons and years to enable later occupancy modeling. Surveys were conducted using Osmos backpack DNA samplers, and field technicians were trained by members of the Hanner Lab. The technicians then followed the survey plan, submitted samples to the Hanner Lab, who subsequently sent data to Zoetica for further analysis. And now I'm really glossing over that step because we'll go into it probably for an hour tomorrow. Our sampling plan included a duplicate every other site, a field negative every third site, and field positives where species of interest were known to occur. In the summer and fall of 2021, 116 sites were sampled and 446 samples were collected and processed for eDNA. As noted, there's been subsequent sampling since, but we're just going to ignore that for now because we haven't worked up the data. This map illustrates the high density of samples planned and achieved in the area during year one over a relatively large area. In some cases, samples could not be collected because locations were not safely accessed or they were unexpectedly dry due to drought conditions in the summer. This highlights the importance of a sampling design like GRITS to account for the realities of the field while maintaining statistical rigor in the results. Our molecular primers focused on fish and invertebrates, and vertebrates in general, and they used the CO1 as well as the 12S regions of mitochondrial DNA. And we'll go into far more detail about the lab methods tomorrow as well. Overall, within this site, after carefully vetting and cleaning data, we identified 1,105 unique taxa, of which there were 644 identifications to species level, 357 identifications to genus level, and 104 identifications to family level. These identifications included many suspected species or species that were noted as important to stakeholders and right holders. So that was a good start because we didn't necessarily have a lot of data from that area. But these data also included some species that were surprises, potentially real species or potential contamination issues that we need to look into. And work is underway to examine these surprises closely. It should be noted that if we had started with traditional surveys alone, many of these surprises would not have been found using traditional techniques. And they've signaled for us to employ additional tools to pick them up in the future. This is a quick look at the number of species detected per taxonomic group. Along the y-axis, you'll see the taxonomic group, and along the x-axis, you'll see the general habitat type. We noticed a pattern almost instantly that we had higher species detections in the fall. We cautioned that some of the sample sizes were higher in the fall because we had some drought conditions in the summer that dried up some of our sites. But still, in many systems where equivalent samples were collected between seasons, the fall detections were still higher. Whether or not this is due to eDNA degradation rates or actual presence of more use of those habitats is yet to be seen. Overall, we also found that the stream river systems and small lakes had the most species detections across most taxa. The primers that we used enabled the identification of a high number of invertebrate species, which you'll see on the very bottom. We were also able to detect mammals, fish, and birds to an acceptable degree. However, amphibians and herpetofauna in general were lower than we would expect for this area, and more work is underway to see whether the primers used can amplify herpetofauna from a mock community from this area. Additional primers may also need to be added in the future. Likewise, other experiments are underway to test the effectiveness of primers for amplifying sequences from known species in the area. Future data collection will include conducting traditional surveys for valuable species to support decision-making and impact assessment. We will also be conducting repeated sampling across years and seasons to support multi-species occupancy models with eDNA metabarcoding extensions. An important point to note is that engagement with Indigenous communities and local citizens is ongoing to share what we're learning, and we're planning to solicit more feedback to include their priorities in our studies. So there are many cautions and considerations. I've listed them here on their slides, and this is not exhaustive. We'll go into a lot of the details on how we consider these things in our workshop tomorrow. But mainly I want to talk a little bit about how this can advance impact assessment and how these data can be used in the impact assessment process. Again, we are very early on in the process, prior to site selection and prior to the actual impact assessment being triggered. However, the use of eDNA and metabarcoding early on in this process is feeding into our decisions about the kind of detailed on-the-ground surveys that we need to employ, both in terms of locations to look or to look harder in, as well as the equipment to deploy, net sizes, things like that, techniques. In turn, the detailed survey data will feed back into our eDNA metabarcoding information, and we'll be able to develop a better library over time. Overall, the combination of these tools, we feel, is leading to a greater confidence in our understanding of the baseline species richness in this area. Ultimately, that will improve our mitigation hierarchy earlier on in the process and lead to a better impact assessment that is focused, has higher confidence, and leads to sensitive and targeted monitoring. We also hope the work will create a large and high quality data set over time that can be used to advance fundamental sciences. A large data set like this will enable the investigation of optimal sampling methods, including understanding the sample sizes needed to reach species saturation, at least in this area and in these kind of habitats. Data may also enable the future tracking of species extirpations over time and advance our basic knowledge of species ranges. Using occupancy models and multi-species occupancy models will help to understand our community assemblages and the ecological drivers underlying them. Finally, large eDNA metabarcoding data sets, along with areas that are well studied using traditional methods, will advance sequence IDs and help for fine-tuning of metabarcoding technologies in the future. So I just leave you with 3 take-home messages before I end this talk. The first is that eDNA metabarcoding does show a huge potential for use in biodiversity impact studies. However, because this methodology is not currently well-established, or doesn't have well-established best practices for use in the IA world, you must be very careful in how it's used. eDNA metabarcoding should be used to enhance but not replace traditional surveys. To an ecologist, it should just be treated as another tool and another way of looking. I'm reminded of the early days of using anabat detectors to detect bat species, where people were really pulling their hair out because we couldn't necessarily tell the difference between certain bat species based on their echolocation patterns. But we could tell that an area was batty, and we could tell that we probably had bats between this size and this size. We knew we had to put out mist nets to catch them, and we knew what size of mist nets to put out. So in a similar way, this is a tool of many that can be used. Second is that expertise, effort, troubleshooting, and resources and time, quite a bit of it, are all needed to create a well-defined and executed program of this magnitude. And third, such studies of this scale will continue to advance applied and fundamental sciences over time, and the usefulness of this tool for these purposes will gradually increase. So this work was a group effort. I'd like to thank communities that provided input into their program, the Hanner Lab at the University of Guelph, especially Bob Hanner and Rob Young, Xu Chen and Nicola Linton, who we've been working with quite a bit. Additional Zoetica staff have also worked on this project endlessly, including Andrea Buckman, Camille Warbington, Celia Chewy, Deanna McKinnon, and Amanda Hamilton. Finally, we'd like to thank the NWMO, and especially Melissa Mayhew, who has been a champion at endorsing this work. We're incredibly gracious to have a client or a proponent that shows a willingness to support such a large and collaborative undertaking. As I said, far more detail about the methodological details and results of this project will be presented tomorrow morning in our workshop. Thank you.